Hi everyone, welcome to our Yikon Genomics next non-invasive PGTA webinar. So today we are so honored to have three speakers here. The first one is Dr. Pai, who is the medical director, Bloom IVF Group in India. And the second one is Dr. Dopozo. He is the founder and chief scientific officer in Argentina. So now I would like to request our Dr. Zhou, our global business director of Yikon Genomics, to share his opinion about our today's webinar. Okay.
Everyone in front of the screen, welcome to Yikon webinar series at YouTube Studio. It is a great honor to have you join us this evening, or afternoon, or morning, wherever you are at the moment, with Yikon Genomics Live to hear about the latest progress from our Nixie user. I'm Dr. Zhou, the Director of Global Business, Yikon Genomics. And Yikon Genomics is very pleased to have you gathering together today and exchanging the user experiences of Nixie, this advanced pre-implantation testing technologies. We are the leading enterprise with rich experiences in providing genetic testing services and solutions for the reproductive medicine field. With the mission for the health of billions in mind, we have helped tens of thousands of families to achieve the pregnancies and having healthy babies. And when we're talking about the IVF and embryo ranking for implantation, as we all know that the morphological indicators are quite commonly used in combination with the pre-implantation genetic testing, PGT, to evaluate and select the most healthy embryos for implantation. However, the morphology of the embryo alone cannot reflect the chromosomal conditions of the embryos. And nevertheless, the PGT itself would also require embryo biopsies to obtain the cells from the embryo for genetic testing downstreams. And this is an invasive procedure for embryos, and its long-term impact is still on the evaluation. And that would shows the value of the non-invasive pre-implantation genetic testing. By detecting the circulating cell-free DNAs present in the embryo culture media, this is allow us to avoid the above issues with no invasiveness to the embryos. And with the aim of letting more clinicians and embryologists like yourself to know about Nixie and how it can help with your patients and improving a better clinical outcomes, we hereby organize this event today to have two of our end users, Dr. Pai and Dr. Zobizo, to share their clinical and application experiences on Nix. It is my great pleasure to introduce our first speaker, Dr. Pai. Dr. Pai is the medical director of Bloom IVF Group and the president elected for the Fogis. He is also the director at the Corporate Affairs and the IFFS, International Federation of Fertility Societies. His research focus is infertility, IVF, endoscope, ICSI, egg frozen, and genetics. Dr. Pai also have had numerous awards, publications, and achievements to his name. He is the fellow of RCOG of UK, and he has been the past Secretary General, Vice President, Chairman for the Fogis, and past President for the ISAR, IAGE, MOG. He was invited to faculties and giving lectures across the world, and he is also the editor of dozens of books and written over 50 chapters so far. and he has been the founder editor of the JGES, the founder chair of India SIG American Society of Reproductive Medicine. Let's welcome Dr. Pai to give the talk titled Non-invasive chromosomal screening, evolution and application.
Good evening, everybody. I'm going to talk to you today on non-invasive chromosomal screening, also called NICS, or non-invasive pre-implantation genetic testing, also called NIPGTA. They are the same, but they are different terminologies. I'm Dr. Rishikesh Pai, Medical Director of Bloom IVF and the President-elect of the Federation of Obstetric Gynec Societies of India, also the Director of Corporate Affairs, International Federation of Fertility Societies, and the adjunct professor, assistant professor at the Eastern Virginia Medical School. I have a lot of interest and I've been doing IVF since 1990. And almost all the newer advances in India, I have been instrumental in bringing them and installing them in the country. It was my privilege and pleasure to visit Shanghai and visit the Yikon genetics in 2019. I was exposed to their high-level techniques of pre-implantation genetic testing, their molecular biology technique, their wonderful lab in Shanghai. And then I came back and established the next-gen sequencing lab in Mumbai, India, and also started doing the non-invasive chromosomal screening or NICS technique as they had propounded, which I had learned in Shanghai. So today my talk is about the NICS. And if you look at the embryo biopsy, the terminology has now changed. The pre-implantation genetic testing is the new terminology. And if you can see here, it's called PGT now instead of the old PGS. So now the new terminology is PGTA, pre-implantation genetic testing for aneuploidy. And normally it entails an embryo biopsy, which can be done at the cleavage stage. When you do a blastomere biopsy, you can see a day 3 blastomere being removed. Then you analyze it by doing a PCR FISH or CGH microarray or NGS, and then you freeze the embryo. You can do a fresh blastocyst transfer if you can get the result quickly, or if you are going to have a delayed result, then you can do what is known as a frozen blastocyst and then transfer after 2 months when the results are available. Initially, it was done for single gene defects. It was called PGT, pre-implantation genetic diagnosis, and now the new terminology for that is PGTM, and subsequently was done for aneuploidy screening, which was called PGS, which is now called PGTA. So you can do the biopsy at the blastocyst stage, as I had told you. It's called trophectoderm biopsy, where you do the removal of the trophectoderm cells. 3 to 4 cells are removed by opening the zona, and then the analysis is done using FISH, PCR, microarray, or NGS. And the blastocysts are vitrified and frozen. And later on, after the report comes back, you can thaw the normal embryo and transfer it. This can also be used in single gene defects or translocation testing, PGTM or PGT for structural rearrangement, and can be used for, as I told you, for aneuploidy screening. It is called PGTA or PGTA for aneuploidy. So the successful blastocyst culture in combination with a laser hatching to open the zona, doing the biopsy and then vitrifying the blastocyst. And the first, all this was possible because of contribution of many people like Dave Gardner, who was the first to do blastocyst culture, Anna Vega, who was the first to introduce laser hatching, Katayama and Koyama, who were also the people who were instrumental in introducing vitrification. So we're combining these 3 techniques of blastocyst culture, laser hatching, and vitrification, we can now do the modern technique of biopsy. So ideal culture continuations for a blastocyst are, you can use a sequential or single media, 5% CO2, 5% oxygen, which is very important because reduced oxygen tension is very important, especially for day 6, day 7 blastocysts. You may have to go for even 3% oxygen, which gives good quality blastocysts. And the nitrogen can be either, if you use a 5% CO2 or 90% nitrogen, or if you use a 6% CO2, then 89% nitrogen. Benchtop incubators, embryoscopes using microdroplet culture is important. And the blastocyst conversion rate varies from lab to lab, means how many day 3 embryos they get converted to day 5 blastocysts. And normally, some labs have fantastic blastocyst conversion rate of nearly 50 to 60%, and average lab has a blastocyst formation rate of around 40 to 50%. Is easy laser hatching trophectoderm biopsy is very important. And if after that, while vitrifying, it is important to collapse the cavity because that gives better recovery when you try to thaw these blastocysts. Frozen embryo transfer is only if the endometrial receptivity is ideal. So when you're going to transfer these embryos, when you prepare the endometrium, and only in an ideal endometrial receptivity, when there is a good progesterone level, good ultrasound scoring, and a good endometrial receptive analysis assay, you can then transfer the blastocysts and get very good results. So what are the indications for PGTA? PGTA can be done for advanced maternal age in women more than 35, 37 years of age. You can regularly subject them to PGTA because the incidence of abnormality is higher in these women. The abnormal embryos which are aneuploid are higher. And so you can do it routinely for women more than 35. The other indication is patient with recurrent implantation failure, which is defined as patient not becoming pregnant after transferring more than 3 embryos with high quality embryos, or the transfer of more than 10 embryos in multiple transfers, the exact number to be determined by each center. And implantation failure is defined as the absence of a gestational sac on ultrasound at 5 or more weeks post embryo transfer. You can also do it for repeated IVF failures. You can do it for recurrent miscarriages, and that is defined as more than or equal to 3 miscarriages, exact number to be determined by each center. It should be noted that patients with a history of recurrent miscarriage have a high chance of successfully conceiving naturally. Patients with abnormal products of conceptus on array or NGS testing, or patients with idiopathic recurrent implantation failures can also be subjected to PGTA. And you can also use PGTA to reduce the time taken to pregnancy, reduce miscarriage rates, reduce the incidence of multiple births using only single transfer of a euploid blastocyst, etc. You can also use it for research, new techniques when you're introducing in the lab, such as in vitro maturation, freezing techniques, spindle transfer, CRISPR, all these embryos that are being created by using modifications in the lab, all these need to be tested for genetic abnormalities, and you can use pre-implantation genetic testing for these techniques. So what are the advantages of trophectoderm biopsy? It is easy to perform. It is minimally invasive because of laser hatching. And less skill is involved compared to a blastomere biopsy. Ease of vitrification enables proper genetic testing using NGS. Frozen embryo transfer can be done only if the endometrial receptivity is ideal. If the receptivity is not ideal, you don't have to thaw the embryo. You can always transfer it in another cycle. And the added advantage of blastocyst transfer in a frozen cycle gives it higher pregnancy rate, decreases in multiple pregnancy rate, decrease in miscarriage rate, and increases the chances of a genetically normal birth. So there are different techniques that were done for analysis. The initially was the fluorescent in situ hybridization, but the problem with it was you could analyze only 12 chromosomes, and this is the biggest problem. All chromosomes could not be tested because of signal overlap, hybridization failure, high error rate, and was demonstrated to be ineffective by Mastenbrook in fertility sterility. And that's why the next technique was evolved, and that was the 24 chromosome test called the comparative genomic hybridization, which was introduced by Yang et al. In 2012. However, there were advantages, but there had the problem of mosaicism was not that great with this technique. So then came the next gen sequencing, and this was introduced by Nathaniel Treff in his landmark paper in fertility sterility, where he had the paper of evaluation of targeted next gen sequencing based pre-implantation genetic diagnosis of monogenic diseases. And then Zhang also in 2013 put a paper on massively parallel sequencing for chromosomal abnormality testing in trophectoderm cells of human blastocysts. And Zi Hong in 2015 have published a randomized comparison of next-gen sequencing and ARIC CGH for PGT screening, a pilot study. And all these showed that the next-gen sequencing was the technique of choice. And you can see here that you can use a next gen sequencing for various purposes. You can use it. You can see here. You can do the biopsy of the trophectoderm by after cutting the zona. You take out 3 to 4 cells. You can do the flipping technique also for taking the cells off. Or you can collect the culture media from the droplets around it, and then these are tube. The tubing is done. So where in a buffer you put the biopsy, either the cells are put or the culture media around the embryo for doing non-invasive chromosomal screening. And once the tubing is done, you then send it to the lab where it is basically stored at minus 20 degrees centigrade. These are the long-term storage of DNA is normally done at minus 80, but the short-term storage of DNA can be done at minus 20 degrees centigrade. And then you wait for enough embryos to accumulate, because you need to, when you are running the test, we normally do 20 embryos running, or sometimes 40 embryos at one time. You can run as much as 90 embryos at one go. But we at least wait for 20 embryos to come before we can run the test, which so it becomes cost effective. So when enough embryos are there, we will take them out and we'll thaw the embryos, and then we will do the amplification using a PCR machine. After 1.5 hours, we have take the amplified DNA out and we do a library preparation. So we tag the embryo and do barcoding of each embryo, and then re-amplification is done for 1.5 hours. So collectively within 3.5 hours, amplification is done, and after that, the template generation is made where all the DNA of different embryos is mixed into single cartridge. Once this is done, and because of the DNA tagging, you can mix all the DNA in one cartridge. So once this is first the PCR amplification, you can see here, is done, and once the amplification is done, then again library preparation is done, and then we put them into the template for the template generation, where the samples are mixed and the DNA is stretched over magnetic microbeads and amplification is carried out. So this is, before we do it, we can test that whether the adequate DNA is amplified or not by using a qubit, a machine, which is automatically tells you. And this is a template generation where all the DNA of different embryos will be mixed into a single cartridge. This takes about 3 to 3.5 hours time, after which once the cartridge is prepared, that cartridge is loaded into the sequencer, and then sequencing starts, which takes about 3 to 4.5 hours. And all the reports are then uploaded on the cloud, where they are read. They can be read directly by the human, or using artificial intelligence, you can read it, and then the final reading can be done by the PhD who is actually doing the test. So this is the template, and once the cartridge is created, then you can put it into the sequencer, and you can do the sequencing, which takes about 4 to 5 hours time. So that's how the next gen sequencing is done. And you can see here that we can do it on various tissues. You can do it on sperm, oocytes, embryos, culture media, endometrium, so blood, amniotic fluid, buccal mucosa, chorion, etc. So the sequencing is done by, so we have if you want to recap the test, a library preparation where whole genome amplification is done, and fragment library construction with iron. We use an iron single sec kit. We can use a Yikon kits as well. Nowadays we are using the Yikon kits. And then template generation is done using Iron Chef. Sequencing is done, and then with the 520, 510, 520, 540 chip, and then analysis is done by nowadays we send it for analysis to Yikon Genetics lab for the final analysis, and then our PhDs read the report. As you can see here that you can read the report. This is a euploid embryo where you can see that at 2 mark, all the chromosomes are arranged here, while there is a single aneuploidy here of minus chromosome 18. 118 is missing. And then we have got complex aneuploidies. You can have chaotic patterns, or you can have segmental aneuploidies. So the advantage is all chromosomes can be tested, next gen sequence advantage. It is a very high resolution, can analyze mitochondrial copy number, high throughput, multiple samples can be tested. As I told you, we can test as much as 90 DNA materials due to the library preparation and barcoding, more accurate in detecting partial aneuploidy, mosaics, and translocations compared to the array CGH. NGS screening does not require co-hybridization of DNA control samples, while RS-CGS testing requires both male and female DNA controls in the hybridization step. Can do other tests such as non-invasive prenatal testing, NICS, expanded carrier screening, etc. Is of course time consuming. Takes about 13 to 16 hours, and fresh embryo transfer. That's why it's not possible. You have to freeze the blastocyst after taking the trophectoderm biopsy, and after the next month or 2 months, you can put it back. It is costly to install and operate, but once you have a lot of turnover of the embryos, then it will become cost effective. So this was a study. It was comparing the NGS versus RS-CGH in a single thawed euploid embryo transfer cycle. It is called STIT, single thawed euploid embryo transfer cycle. Friedenthal et al. In Fertility Study 2018, and they compared the two techniques of NGS and array for trophectoderm biopsy, and they showed that NGS improves the pregnancy outcome, has the ability to identify and screen for embryos with reduced viability, such as mosaic embryos and those with partial aneuploidies or triploidies. Pregnancy outcome with NGS may be improved due to the exclusion of these abnormal embryos. So what are the disadvantages of the trophectoderm biopsy? The equipment is costly. You need a micro manipulator, you need a laser, need skilled manpower. It is very invasive and may cause short-term embryo damage, resulting in decrease in the implantation rate and pregnancy rate. You need a very good embryologist. Otherwise, he may damage the embryo in the process of biopsy. There are implications of long-term damage. We don't know what maybe the long-term damage is when we are opening the zona and then taking the 4 to 6 cells out of the trophectoderm. And then there are the problems of mosaicism resulting in false positive and false negative rates. So if you look at the embryo, the 4 types of embryos are there. These are the euploid embryos. These all are gray cells, all are normal. If there is less than 30% abnormal cells, we classify it as a euploid embryo. On the other hand, if there are more than 70% abnormal cells, the red cells, we classify it as an aneuploid embryo. In between these two are the low mosaic and the high mosaic. So an aneuploidy where there is 30 to 50% cells are normal, abnormal, we call it low mosaic, and 50 to 70% are called high mosaics. So the mosaicism rate in the embryo population is 5.8%. Normally, you can definitely diagnose whether it's a euploid or an aneuploid embryo. And 3.8% low mosaic and 2% of high mosaic is generally seen as per Santiago Munoz study of 2017, where he studied using egg donors as a control population and have demonstrated that the rates of resulting embryo euploidy differ between IVF centers by as much as 40%. So what is the mosaicism problem? So here you have got a normal, the abnormal embryo, the inner cell mass is the area of the embryo which gives rise to the child, and the outer trophectoderm is the area from which the placenta is formed. So we normally take the biopsy of the trophectoderm. So when you, in this patient, for example, this is an abnormal embryo, but if you take a biopsy, you will think that it is normal. So it is called false negative. Actually, it is a abnormal embryo, but your result is showing normal. And in this case, where the actual embryo is normal, the inner cell mass is normal, but the biopsy has abnormal cells, you will call it a false positive. So the embryo is biopsy is positive, but actually it is a false positive. The embryo inside is normal. So what are the problems? If there's a false negative embryo, means an actual abnormal embryo, but report has come as normal, the false negatives are of concern because that transfer may lead to either no pregnancy, miscarriage, or worse yet, an abnormal fetus. So if this embryo by mistake, we think that it is normal and we transfer this embryo back, you may think that it may not implant or there may be a miscarriage, or if it continues till term, you may have an abnormal child. Or on the other hand, if there's a false positive, are particularly concerning as this may lead to discarding embryos that would otherwise lead to normal birth. So in this patient, you may think that it is abnormal and you may discard the embryo, but actually the inner cell mass is normal. So these are some of the problems of normal false positive and false negative. And mosaicism policy is there of different groups. So embryos that have another full chromosome abnormality in addition to the mosaic finding will never be reported as mosaic and will not be transferred. Embryos with mosaic findings in the following chromosomes will never be reported as mosaic and hence not transferred. For example, trisomy 13, 16, 18, 21, X and Y trisomies will never be transferred. So this is as far as the mosaic policy is concerned, but everyone has their own mosaic policy for transfer. And if you look at this paper of fertility sterility, Perti et al. In 2021, he had a paper rate of 2 recurrent implantation failure is low, results of 3 successive frozen euploid single transfer. The live birth rates after the first, second, and third frozen embryo transfer in their group was 68, 64%, 54%, and 54% per transfer, respectively. The cumulative live birth rates after up to 3 consecutive frozen embryos, single embryo transfer, where the blastocyst that was put back was normal, was as high as 92%. So this technique of testing the blastocyst and putting the euploid blastocyst back has a fantastic pregnancy rate. The miscarriage rate was, as you can see here, was 7% in the first transfer, 8% in the second, and 12.7% in the third. So an average of 8 to 10% was a miscarriage rate in patients where frozen embryo transfer was done on blastocysts that were tested for aneuploidy, of course, by the technique of biopsy. So this was the STAR trial by Santiago Munich. A total of 661 women were done, and he randomized it into 2 PGTA and non-PGTA. Only morphology was used for selection, and in half the group, the genetic testing of the trophectoderm biopsy using NGS was used for selection, and they found that the pregnancy outcome was similar in both arms in women less than 35 years of age. But in women more than 35 years of age, there was a 14% increase in the pregnancy rate in the arm which was tested for genetics as compared to the morphology arm. And the miscarriage rate in both the arms was around 10%. So this is very important study, as you can see here. So are there any alternatives to the gold standard of pre-implantation genetic testing? So we have the PGTA as a gold standard. Everyone has accepted it as a gold. So is there any alternative which is a test which is non-invasive, simple, easy, no skill required, less costly, good sensitivity and specificity, good prediction of euploidy and implantability? So there are different approaches to achieving high pregnancy rate, low multiple pregnancy rate, low miscarriage rate, low genetic abnormality rate. So we can do this by morphological analysis, which we do it normally, or time lapse, or we can do with a time lapse in combination with the artificial intelligence, or you can do a standard photograph with the AI, or you can do the non-invasive chromosomal screening of DNA in span culture media. So there are different things that can be done for this thing. So different people are using it. So some of them are doing blastocyst culture and putting one blastocyst at a time. So first time they put the blastocysts in fresh transfer and remaining are frozen, and every time they put a frozen transfer based on morphological criteria. So majority of the world are doing that. In United States and China, they are doing blastocyst culture and PGTA. You can use it by doing array or NGS. And some of us are now doing blastocyst culture with selection that is based on either morphology or time lapse or photo micrograph with artificial intelligence or non-invasive chromosomal screening of span culture media. So these are the various methods. So one is a time lapse incubation, which is being done quite often now. And many people are combining time lapse with PGTA, or you can combine time lapse with non-invasive PGTA as well. And this is the Trebinski et al. In RBMO 2017 did a meta-analysis of 5 randomized controlled trials, and it showed that the application of time-lapse monitoring was associated with significantly higher ongoing clinical pregnancy rate, significantly lower early pregnancy loss, and a significantly increased live birth rate. So time-lapse, according to him, has definitely improved all these parameters. However, there is a disadvantage. It's a very costly incubator. The outcomes are not proven. There is a damage, there is a light exposure. Every time you take a photograph, every 15 minutes, there may be some amount of light exposure which may query damage the embryo. There is insufficient evidence of difference in live birth rates miscarriage rates, stillbirth rates, or clinical pregnancy rate to choose between the time lapse with or without embryo selection software and conventional incubation. This is what Armstrong and et al. Group had shown in Cochrane review of 2018. So you can see here that the eggs, after they are injected, they are put into the wells and they are covered with oil. You can see these eggs one at a time, they are put in the wells covered with oil, and then we can. So in our unit, we not normally use time lapse, and we can, that same well can be used for collecting the culture spent culture media for non-invasive testing as well. So we are using time lapse to select. So if the time lapse shows a blastocyst, but the development of the blastocyst was abnormal, then you don't have to use the NICS on that on that embryo. So you don't have to do PGTA, non-invasive or invasive PGTA if the time lapse has already shown an abnormal fertilization. So you can see here that by looking at the video, see, this is a fertilization where there is a pronuclear overlap. Then the first cleavage is there, 2 cell, 4 cell. Then we have the 8 cell on day 3. And then we have the morula, and then we have the cavitation, and finally the expanded blastocyst. So all these things can be seen, and the timing of cleavage can be used for analysis of the best blastocyst, as well as abnormal fertilization also can be analyzed. So if there's an abnormal fertilization, then that time lapse can be eliminated. That embryo may not necessarily be subjected to PGTA or non-invasive PGTA or the NICS as we call it, because it is a waste of our resources. So time lapse can be used in combination with PGTA to select the best embryos for analysis. So of course, you can also now use artificial intelligence, and there is a new score which is known as IDS score, which is based on analysis of 150,000 embryos using a convoluted neural network. And you can use this to actually predict which is the embryo which may be euploidic and which will have the best chance of implantation. This was a study by D. Tran et al. In 2019, Human Reproduction, where was a study of analysis of 115,000 embryos, and they used it to prioritize the transfer. So best embryo was selected, and that was transferred first, and they showed a very good outcome using the time lapse. You can, but the time lapse is very costly, so you may not have that in your unit. So instead of time lapse, you can use other artificial ranking systems. In our unit, we also have got ERICA, which we use for ranking the embryos using a inverted microscope with Hoffman modulation contrast optics and a high definition camera. And on day 5 or 6, when the blastocyst formed, we take the photograph, we export it to the server, and the server analyzes it based on 25 million manually trainable parameters. It's a cloud-based technology and gives a 80% accuracy of the predicting the best blastocyst for transfer. So you can actually take the photograph and upgrade it, upload it, and you can have a system where ranking can be done. Again, ERICA can be combined with non-invasive testing, and you can get a very good combination and predict a very good outcome. So non-invasive testing of the embryos using NICS technology in combination with ERICA or similar artificial intelligence, visual technology, or even morphology together can predict a very good outcome. So in our unit, we are getting a lot of pregnancies with ERICA as well. So in 2016, as I told you, PGTA was the invasive technology. So in 2016, Zhu et al. Published this paper of non-invasive chromosome screening of human embryos by genome sequencing of embryo culture media for in vitro fertilization. And they used a multiple annealing and looping based amplification cycle called MALBAC for whole genome amplification. Next gen sequencing was performed on the spent culture media used in culture of human blastocysts. It was done on 42 blastocysts, and results were validated by performing standard profectoderm biopsy with an NGS. This is known as a concordance study. With this validated NICS method, chromosomal screening on IVF embryos from 7 couples with balanced translocation, azoospermia, or recurrent pregnancy loss was performed. 6 of them achieved successful clinical pregnancy, and 5 had already achieved healthy live birth. So when I read this paper, I started communicating with the Yikon genetic group, and I went to Shanghai, and I got trained in this technique of non-invasive screening. So how does this work? You do the oocyte retrieval, then ICSI, then you do the culture from day 3 to day 5. You can freeze the embryos and then thaw them and culture them. But nowadays in our group, we are culturing them, even fresh embryos, we are culturing them till day 5. And then we are collecting the spent culture media. normally we wait for the blastocyst to expand. So in the initial phase, we are giving a laser cut, but nowadays we don't give a laser cut. We wait for day 5, from day 5 to day 6, based on Catherine Rakowski's group's work. We wait for the blastocyst to expand and hatch. At the time of hatching, that is the time we pick up the culture media. In those patients that the blastocyst doesn't hatch, we can enable the blastocyst to hatch by giving a laser cut on the zona to help them to hatch. And if they still don't hatch, then we can do a standard biopsy on this patient. So we counsel our patient that if suppose we are doing a non-invasive testing and a particular blastocyst doesn't hatch, we may have to do a standard biopsy, because we have found that in those patients where the blastocyst is not hatching, the amplification dropout, DNA dropout amplification rate may be higher. So we do, and then this is the standard NGS sequencing is done and report is done. So the advantage is that there's no biopsy and only spent medium is needed. No specialized instruments are needed. Avoid sample loss and high detection and success rate, and can detect 1 to 2 picograms of DNA. So this is based on the Malbach multiple annealing and looping based amplification cycle. Malbach whole genome amplification involves 5 cycles of quenching, extending, melting, and looping. Unlike conventional DNA amplification method that are non-linear or exponential, in each cycle DNA copy can serve as a template for subsequent cycles. Malbach utilizes special primers that are 8 variable and 27 flexible fixed nucleotide primers, and these allow amplicons to have complementary ends and therefore to loop, preventing DNA from being copied exponentially. This results in amplification of only the original genomic DNA and therefore reduces the amplification bias. So you can see here that there are 5 cycles of looping, and followed by that there's a standard amplification. So if you look at the concordance rate, what is the concordance rate? So you take an embryo, you take the culture media around it for the DNA and you amplify it. You also take a biopsy of the embryo, and then you can also do the analysis of the entire embryo, or the inner cell mass, and then you see whether all these things are corresponding or not. So there was a paper by Huang et al., and he showed that the cell-free DNA concordance rate with DNA of whole blastocysts reached 93.8%. This result strongly suggested that embryo cell-free DNA is robust and can be more representative of the embryo chromosomal status than profectedom biopsied embryos. In a recent study obtained, a concordance rate of cell-free DNA with inner cell mass was similar to those of profectedom biopsy and inner cell mass, 87.5% as compared to 84.5% respectively. This was Rubio's paper in American Journal of Obstetrics and Gynecology 2020. Taken together, these findings instead supported the hypothesis that cell-free DNA secretion is a physiological phenomena of each embryo, independent of their quality and ploidy status, and not related to self-correction by apoptic events. And these are the various studies. You can see that there was a learning curve, and as you went along, different authors have shown the concordance rate has gone up, and we also have around 80% plus concordance rate now, and it's becoming better and better as we go along. Does day 6 blastocyst, so this is very important, does day 6 blastocyst transfer in NICS decrease pregnancy rate? An objection to NICS is that an expanded culture till day 6 is required to obtain sufficient cell-free DNA quantity for robust results. And Shear et al. In RBMO 2020 has shown that evaluating the effect of day of cryopreservation showed that in frozen transfers of euploid embryos assessed by PGTA, the day of cryopreservation did not significantly affect live birth rate. So remember, the day, even if it is a day 5, day 6 expanded, you wait till day 6 for the expansion and hatching, and you then collect the cell-free fetal DNA, cell-free DNA, you freeze these embryos, and then, but when you are transferring them, you must transfer them on day 5. So even if you freeze the embryos on day 6 or day 7 or day 5, you can freeze them without damaging the embryos, but you should transfer them on day 5 of when you are doing a frozen embryo transfer cycle. So what are the advantages of NICS? Non-invasive, easy, no equipment or training needed to collect sample, no problem for sample loss, can detect very low levels of genetic material, is comparable and compatible with the two main platforms of Illumina or Torrent. It assesses the implantation potential instead of the aneuploidy status. It is a grading technique rather than a diagnostic technique. It cannot replace PGTA, rather is supportive to PGTA. And future strategies is to combine PGTA and NIPT to more accurately choose the embryo. So if there are studies where they have shown that if you do a PGTA, means actual biopsy, and you do a non-invasive testing of the culture media also, and if there is a concordance in both of them, so the embryo is duploid both on biopsy and the culture media, that embryo gives the best pregnancy rate. So this is maybe the future will be that we'll combine both the techniques. So Rakowski in Fertility Study 2021 had put out a wonderful paper on the advantages of NIPGTA. And she said that available evidence has indicated that the performance superiority of NIPGTA over that of invasive PGTA, and therefore greater reliability of NICS for the determination of embryo karyotype. The STAR trial revealed a limited age-dependent benefit of invasive PGTA. In invasive PGTA, because of the exclusion of poor quality embryos not suitable for biopsy and the possibility of embryo damage during the biopsy itself, there may be a problem of sample loss. This is effectively overcome by NICS, and there is no need for embryo biopsy training, and medium collection is easier to standardize in NICS. So these are certain advantages. And so what is our approach? So robust counseling is very important. So you have to tell them that this is a non-invasive test. It is a grading ranking test rather than a actual diagnostics test, and that we are not going to discard the embryos which are not normal on NICS, because there's a high chance that those embryos may still be normal. So we would not discard these embryos. If at all we have to discard these embryos, then we would definitely do a biopsy, be sure, and then only discard the embryo. Proper dissection of cumulus cells is important to prevent maternal contamination. Day 5, day 6, expanded blastocysts should be targeted. If blastocyst does not expand, laser cut is given to assist hatching. If still the blastocyst does not expand, traditional biopsy is performed, or the procedure may be cancelled if the patient doesn't want to do traditional biopsy. She says, no, I don't mind doing a morphological based transfer, or I don't mind doing a morphological and artificial intelligence based transfer, then we can go for that. Blastocyst should be cultured in 20 microliters of media. We prefer the Queen's media. Day 5, 6, frozen blastocysts are transferred on day 5. So as I told you, if the blastocyst is frozen on day 5 or day 6 or so very rarely on day 7, if you freeze them, but when you are doing the transfer, you will do it on day 5 after starting the progesterone. Failure of DNA amplification can be compensated by thawing and biopsy of blastocysts at no extra cost. So suppose we do the collection of the spent culture media and the DNA doesn't amplify, then we can counsel them right at the beginning that if there's a failure of amplification, we can do the thawing of the blastocyst, we can do the biopsy, we can refreeze the blastocyst and do the analysis, but we'll not charge you for it. Discarding of blastocysts after appropriate consent, and offer to biopsy blastocysts and perform PGTA prior to discarding. So this is very important from the medical legal purpose, that we tell them that, okay, we are not going to discard because there is a possibility that these blastocysts may be normal. So we will take a biopsy also. And even if that is abnormal, then we may discard. So this is how you can protect yourself from medical legal litigation. So in our patient range, we have started doing it on a clinical practice. Almost all patients that we have done are more than 2 failures till 7 failures. All ICSI, we do fresh frozen embryos going from day 3 to day 5. As I told you, as the blastocyst is expanding from day 5 to day 6, that is the time when we collect the spent culture media for cell-free DNA. Blastocyst decompression is done prior to freezing, and we normally we have had babies now born with this technique. If you look at our initial results now, we have done 230 embryos with non-invasive testing, 79 patients, and our quality has failure of QC is only 6.6%. And so now we have started getting the hang of it. The normals were around 38%, abnormals were 35%, and mosaics were 19%. So there is a good now correlation. And if you can see here that out of the 89 samples, total patients 80, we have done 30 embryo transfer cycles with a positive pregnancy rate of 70% and an ongoing pregnancy rate of 63%. We had only 2 miscarriages. Our miscarriage rate is around 9.5%, which is corresponding to Santa Muniz paper. If you look at the ASRM guidelines of transferring embryos, if patient of any age, if you have a euploid embryo, you can transfer only one blastocyst. Less than 35, you can do a single embryo transfer again, irrespective of the embryo stage and irrespective. But if between 35 to 37, again a single transfer. 38 to 40, more than 3 cleavage or 2 blastocysts. But if you have a tested embryo, again a single blastocyst transfer. So more than 4 cleavage stage embryos or 3 blastocysts for more than 41 to 42 years. Again, if you have a tested, genetically tested embryo, you don't have to do more than one transfer. So in conclusion, blastocyst culture may be the preferred way for all cases. Single embryo transfer using non-invasive testing may be done, resulting in decrease in the time taken to pregnancy, decrease in the multiple pregnancy rates, and increase in the implantation rates. This can be offered to first time or second time embryo transfer patient. Non-invasive PGTA or NICS may be done keeping in mind that we are more focusing on the implantability of the embryo rather than on its genetic status. Grading and selecting best quality embryos using the invasive method of trophectoderm biopsy and PGTA may be ideal in patients more than 35 years of age or more than 2 RIF or more than 2 miscarriages or with abnormal embryo. We can also do it. Now we are offering that grading for patients with 2 recurrent failures. But when there are 2 miscarriages or more, I'm still offering them the invasive technique of the biopsy. We don't offer them the, so for recurrent miscarriages, I would still go for the invasive technique because it's more traumatic for the patient. But for recurrent implantation failures and patients who are more than 35 who have not had a child, I would go for a non-invasive testing. As we evolve, even this can be replaced by non-invasive testing. So once we are confident, as we go along and become more and more confident of our technique, of our amplification, etc., of our results, and more studies come in, we can then go totally towards non-invasive testing. In these patients, time-lapse may be used to identify embryos to be included. So time-lapse can also be used in combination because if there is an abnormal fertilization, there is a 3 pronuclei, then we don't have to use that embryo for testing. We are planning to offer a combination of PGTA and NICS also in the future. So if patients are there, we will say, okay, we can do both. We can not only do a biopsy, but we do the NICS as well. And if there is a euploid, that's the best embryo for transfer. Remember that most of us had successful IVF practice without doing PGT, as we all consider this as just an add-on. It is better to have a non-invasive safe technique with a concordance rate of 80% rather than not having an invasive technique. PGTM should be done by the traditional methods of PGTA with trophectoderm biopsy and genetic analysis. So in PGTM, I would still do, although I know that Yikon Genetics is now evolved a very good technique with using linkage analysis, and they are doing PGTM also in the non-invasive way. However, it's a learning curve, and I'm sure that we will learn that technique as well, and slowly go over to the totally towards the PGTM also for in the non-invasive way. However, recently non-invasive PGTM is being effectively carried out using linkage analysis. As I told you, the future would be a combination of time-lapse incubation, artificial intelligence, non-invasive chromosomal screening, and next-gen sequencing for implantation window with a single embryo transfer. So we can do also NGS for implantation window using array. So we can do what is a selective embryo transfer, minimizing miscarriage rate and maximizing the chances of a genetically normal infant without any imprinting disorders. I would like to leave you with this photograph of 2 iconic ladies now who have discovered the CRISPR and who got the Nobel Prize for Chemistry in 2020 for their great and breakthrough discovery of CRISPR, Dr. Emmanuel Charpentier and Jennifer Doudna. Thank you for a very patient hearing.
Thank you for your excellent talk, Dr. Pai. This presentation touched upon all the techniques briefly for the embryo screening, and he also talked about how they have evolved over the decades. By comparing the conventional biopsy-based PGTA and non-invasive PGTA, and advanced technology such as time-lapse and AI in embryo morphology grading, etc., I would like to highlight his successful experience with NIXI and in combination with the AI plus NIX and the NGS. We can achieve a better pregnancy rate without genetic abnormalities. Our next speaker is Dr. Dopaso, the founder and the CSO of Biocordis. Biocordis is a company of genomics and bioinformatics specialized in human health, with a focus in reproductive and oncology in Argentina. Dr. Dopaso has been focused on the genomics research since his postdoctoral period, and used to be the professor at the University of Buenos Aires. He also has extensive professional expertise in bioinformatics, and in the past few years, Biocordis has been working with over 14 IVF clinics across Argentina and Uruguay, developing PGT and NIPT, and other genomics studies for human health. Let's welcome Dr. Dopaso to give us his talk, Non-invasive pre-implantation genetic testing for aneuploidy.
Hello everybody, my name is Hernando Paso. I am the scientific director and founder of Biocodices, a genomics and bioinformatics company for health in Argentina. Thank you very much, Yikon Genomics, for this invitation. In the following minutes, I will do a brief overview on non-invasive technology for the genetic study of embryos, human embryos. And we will show the results we obtained in a trial carried out entirely in our country. Finally, I will comment on the advantages and disadvantages of the non-invasive pre-implantational genetic testing for aneuploidies. Pre-implantation genetic screening has a history of approximately 3 decades. During this time, the technology were succeeding, incorporating improvements and evaluating an increasing number of markers, and transforming mainly analog data to digital data. Among the initial technologies, we can mention qPCR, fluorescence in situ hybridization, array comparative genomics hybridization, and finally NGS, next generation sequences. As you can see in the picture, the improvement have been enormous from the point of view of the number of markers used, for a dozen in the very beginning, located in very specific regions on the chromosomes, to millions of markers distributed throughout the human genome. In 2017, the terminology of pre-implantation genetic screening and diagnostics also changed to PGT, ASR or M, meaning aneuploidies, structural rearrangement, Mendelian, PGT. We reach no doubt about what each type of test does. The key for bringing all these genetics improvement to the clinic was the embryo biopsy. This technique is performed basically using a laser. Of course, it's an invasive protocol, and of course, it's necessary before the development of any techniques, of molecular techniques. However, the biopsy possibilities are not general for all the IVF clinics. That is, as I said here, the requirements are different. You need a trained staff, specialist equipment indeed. And of course, we discussed today if there are some consequence about the biopsy itself in the development of the embryo. However, non-invasive pre-implantation and genetic testing for an embryo drastically changed this scenario that was fixed basically during the last 30 years. Of course, there are other studies, yes. Here I mention only two. That is the probability or the possibility of that biopsied embryos that develop preeclampsia and placenta previa. And another interesting studies that say that the genetic representativeness must be basically do using more than 10 cells, 10 cells biopsied cells. That is basically impossible to obtain this kind of material. Then from 2014, as we will see in the next slide, the cell-free embryonic DNA is gaining more and more impetu, and basically we have here the first studies in 2013 that look at the cell-free embryonic DNA as a proxy for embryo viability. That is, there was an obvious representation of DNA in this in the culture media, and was initially, as I said, interpreted as a case of fragmentation of the embryo. Here we say, for example, different initial research doing in 2013, where the mitochondrial DNA content in embryo, say the title, is significantly associated with human embryo fragmentation. That is, no one thinks that the cell-free embryonic DNA has a normal process of the biology of the embryo. There are suspicions that it could be not be a normal process, but a pathological process, of course. After that, the study of pathological Mendelian variables in the DNA of the culture media continued during 2013 and 14. Here we have different works using this DNA for the study of alpha thalassemias and or X-linked disorder in human embryos. However, in 2016, publication began to appear asking whether the PGS is a valid technique with this discarded material, without the need to use the trophoectoderm biopsy. From 2016 to 19, the publication will show successive improvements in yields, decrease in errors, contamination, and the statistical value that lead to think that it's not only an effective method, but that it can offer better results than trophoectoderm biopsy results. In 2021, this year, a study carrying the major study, yes, was carried out with more than 1,300 embryos, and subsequent publications continue to confirm the usefulness of the non-invasive PGTA as a diagnostic method that can be replaced invasive methods on the embryos. The basic protocol of NI-PGTA consists basically of three different steps that probably could be done in different workspaces or different labs. It depends on the technological capacity of the clinics. The first is is run in the embryology lab. We're coming from the fresh oocytes, the fertilization using ICSI is demanding, and the culture of the blastocyst to day 6 or day 5, it depends on the quality of the embryo, and the collection of the culture media in day 6 or 5. Yes. The second step runs in the genomics laboratory, coming from the amplification of the DNA in this collection media, the preparation of the NGS library, and finally the sequencing in different machines of NGS. It depends on the company. It could be Illumina or Thermo Fisher or wherever you have. The third step is the data analysis itself. Here we have decision making on the level of resolution of a nucleotide that we need to think or to do, and finally the generation of reports. Of course, the Chrome Go tool that is available on the web of the Yikon Genomics is a service that you can use for these final steps. Then here we have perhaps the more difficult part of this technique is the first one, that is the collection of clean DNA in the culture media. That is the collection of the enough DNA that can be effectively amplified to make a good report is of course very, very important, and to eliminate the possibility of contamination from the maternal cumulus cells or contamination of the culture media that is another demanding part of the techniques. The success and accuracy of the NI-PGTA depends on the number of different factors. Here I mention only 5, but I only dedicate time to the first third. Basically, we have the first one is the careful application of DNA collection protocols. Of course, you can find this in the Yikon Genomics website. But here you can two different kind of protocol. One is the collection of the DNA from fresh embryos, and the second protocol is from the thawed embryos. In the first case, yes, from fresh embryo, you must to denudate the oocytes. In the second step, you must to fertilize the embryos using ICSI techniques, and the culture up to day 3. After that, you need to change the medium from in the day 3 to another capsule until the day 4. You need to wash in day 4 and transfer to the new culture medium the embryos. The cultivation of the embryos until the blastocyst develop to a morphological grade 4 BB is very, very important. And as our experience is that here the step number 4, that is to wash in day 4 and transfer to a new culture medium, is critical. Yes. Finally, you collect the culture media in a tube and move to the second step in the genomics lab. In the case if you have thawed embryos, you need to wash after devitrification and recultivation for at least 12 hours, and the collection of the culture media after that. Of course, here in the second is a second point I mentioned, to avoid the maternal contamination. For doing that, in order to do that, you must to remove all the cumulus cells in different washes. That is, I mentioned here the different steps of washes where you change yet the embryo in different capsules, and this is a key factor for a fair determination of the copy number and sex of the embryo. Finally, the third step is the whole genome amplification. Here you have the Malvac product that is provided by Yikon Genomics. Malvac means multiple annealing and looping based amplification cycles. That is a very, very clever way of to do a PCR. And in these steps, you will have a lot of DNA to prepare the libraries, the genomics libraries. Of course, there are another two that is genomics platform you use. That is, as I say, Thermo Fisher, Illumina machines. This is irrelevant, basically. For the methods, we use particularly Illumina, but it works very well in the two kind of machines. And finally, the application of artificial intelligence is very important for the analysis of data. All these steps of the point number 5 is provided by the Chrome Go platform by Yikon Genomics. In the next slides, I show you one of the most interesting works that compare the non-invasive PGT with the complete embryo and the trophectoderm, that is the invasive methods. Here, this work was done by the group of Yikon Genomics and Katerin Rakowski. And the title is very, very interesting. Non-invasive pre-implantation genetic testing for an aneuploidy in a spent medium may be more reliable than trophectoderm biopsy. Here is very, very interesting because they work with this kind of comparison that is coming from ICSI embryos, yes, that were biopsied previously. Yes, they know the result of euploidy and euploidy sex, etc., of these embryos. They compare these embryos and the whole embryos, yes, that was taken as the gold standard, with the collection of the non-invasive PGT, the collection of the culture media in the comparison. Then they have two comparisons: the embryo versus the trophectoderm biopsy genetic features, and on the other hand, they have the complete embryo result and the result from coming from the culture media, this non-invasive PGTA. Then these two comparisons finally are compared here in these two columns. Here we have the different performance, yes, or values of performance. That is, false positive rate, false negative rates, positive predicted value, negative predicted value, sensitivity, specificity, and the percentage of concordance for euploidy, for euploidy, and the percentage of concordance for chromosome copy numbers. Then, as you see here, basically the first column for non-invasive versus the invasive, that is trophoectoderm biopsy, compared in both cases with the complete embryo, you see that all the values are better in the first column, yes, that is non-invasive, than in the second one. That is, for false positive rate, we have a 20% in non-invasive and 50% from invasive. False negative rate is 0 in both cases, but its positive predicted value is 91 for non-invasive and 78 for invasive. And for example, the specificity or sensitivity is 100% for both, but the specificity is 80% for non-invasive, but is 50% for trophoectoderm. And in the case of concordance for the ploidy of the embryo, we have almost 94% versus in non-invasive versus 82 in invasive of trophoectoderm. That is, as the papers say, the DNA derived from the culture media is more representative of embryonic DNA than the derived from the trophoectoderm biopsy. Basically, these are exactly the results that we can see in these two columns. Therefore, the non-invasive PGTA could be provide a more precise genetic representation of the complete embryo and therefore reduce the bias produced by the mosaicism of the few trophoectoderm cells that probably lead to a false positive or false negative results in comparison with the internal mass cells, internal cell mass, sorry. Okay, then, in the next slide, I show you what we did in the few months before the beginning of the pandemic. We decided to work together with the embryology laboratory of Procrearte, that is one of the biggest company of IVF here in Argentina. The lab is led by Ariel Ahumada and Francisco Leocata, and they basically work doing the first part, the first step from the culture media, for the oocyte and the ICSI and the culture of the embryos, and they collect the culture media according to the protocols. And we decide to work in a double-blind trial between Procrearte and Biocodices. Basically, we use exactly the same framework of the Catherine Rakowski and Yikon Genomics paper, but the difference is that in this case, we make the libraries for the trophoectoderm using the products of the Illumina. That is exactly what we previously work with Procrearte company or clinics. Then we have results using our previous gold standard, that is libraries using Verisec from Illumina, and we compare this result from that are invasive from the non-invasive libraries coming from the Yikon and Malvac product from embryos, so exactly the same embryos. And we compare the results, between the invasive and non-invasive protocols. This was the first trial fully developed in Argentina between Procrearte and Biocodices. Here I show you the results of that, or the framework we work in the lab, in the genomics lab. Here you see the the first cell-free embryonic DNA quantification in the first run in October 2019, where basically 100% of all the culture media were amplified without any problem using Malvac. We ran our libraries or sequencing using the MySeq machine of Illumina, and as you see, we have 1 million reads, each one of the 55 per basis. These reads had produced a coverage of 1.5% of the gene of the human genome, and the quality were very, very good. That is, 90% of the reads has a Q30 or higher than Q30, and the FASTQ files were uploaded to the Chrome Go tool. The output file of the Chrome Go has a lot of metrics. One of the very important metrics, the coefficient of variation around, in this case, the number of the copy number. In this case, each of the chromosomes has a copy parameter of 2, and the P and the Q arms show a variation around the 2, and in this case, you see that is an X and Y. There is a male, and the coefficient of variation is very, very smooth around the copy number of 1. Of course, you can see each one of the chromosome alone, yes, with all the dots that come from different reads, and you have a lot of information of each one of the metrics. Of course, the artificial intelligence you apply in the Chrome Go characterize the embryo of an A, B, and C from better to worse. And finally, the results we have with the 30 embryo we ran with Procreate, we show here that most of the embryos are or in the diagonal, yes, of this table. That is 17 were aneuploids are aneuploids in the gold standard of the trophoectoderm and the non-invasive protocol of the culture media. 9 were euploids in both methods, and only 4 were euploids in the culture media, but were characterized as aneuploid in the trophoectoderm, that is invasive method. This is not new. All the comparison doing by different authors and published in different journals has this kind of result. What's interesting is that we obtain 87% of concordance, and this is a concordance that is very, very good. It's comparable with the best result of other international groups. And of course, as you see here in the table, you have the comparison of trophoectoderm with different numbers and different publications. But of course, there are another works in the comparison of non-invasive with polar body, blastocyst fluid, and complete embryos. The comparison of the complete embryos were always are higher. Yes, the complete embryo has a lot of DNA. Is better than the comparison than the trophoectoderm alone. But in any case, we are very, very happy with our results. And just for finishing, basically, I will show you 2 tables that I get from this paper that is from Catherine Rakowski, Carlos Simon, too. That is, there's a very, very interesting comparison of pros and cons of the method. But what is interesting to understand in the table 1 and in table 2 is that they show the results of 4 different studies. That is a meta-analysis comparison in 2 different tables. Using 4 different combined studies. In both cases, you will see in these tables that the non-invasive PGTA is more reliable than invasive PGTA in all components of the comparison. That is, in the performance of the performance of the test were better in the case of table 1. That is, the false positive rate for the non-invasive is lower, yes, in non-invasive than in the invasive. But in the case of the table 2, you have the difference with table 1 is that the difference is the day where you collect the culture media. This is very important point, or very important data. Yes, if in the table 1 you have the you collect the medium, yes, from day 3, 4, 5, 6, 7, whatever the day. But in the case of table 2, you have only the DNA coming from day 5, 6, and 7. Then, if you compare table 1 and table 2, you will see that the non-invasive in table 2 has a better values. That is, the performance of false positive, false negative are better in non-invasive than in the invasive methods. That is, all the results using meta-analysis of each one of the methods, must conclude that it's a very, very robust method. And here we have the last result that I have the first Argentinian child born using non-invasive PGTA. This is the birth of the healthy girl in March of this year after 39 weeks of gestation due to uncomplicated delivery. This was headed by Gustavo Martinez and all the group of medical doctors of the Fertilis in vitro clinics. We did basically the genomics results, and in this case we have two different. In this slide, I show you only two different karyotypes. The first one is exactly the karyotype of the girl, and the second karyotype, was not transferred because they show, or these show mosaicism of the Q arm of the 16 chromosome. Then, for finishing, I say that for concluding, pros and cons of the methods of non-invasive PGTI. Here I show you perhaps the first is that we have different evidence supporting the hypothesis that cell-free embryonic DNA show a better representation of the whole embryo or the internal cell mass than the trophectoderm biopsy cells. Standardizing the culture media collection among labs is easier than the standardization of the trophectoderm biopsies in each of the clinics. No embryo biopsy training nor special equipment is necessary for non-invasive pre-implantation genetic testing. And another interesting result, I showed no result about that, is that the non-invasive from towed embryos usually report higher proportion of euploids. This is the case of New Hope Fertility in New York that was published in a meeting last year. And the cons, perhaps the main cons, is that the contamination due to either the culture medium constituents or the culture cells are the biggest weakness of the non-invasive pre-implantation genetic testing for aneuploidies. And of course, finally, a clinical validation study that analyzed the proportion of ongoing pregnancies and live births has yet to be carried out. And finally, to conclude, I just want to thank the group of embryology and medical doctor of ProCreate Fertilis, and of course the staff of the Yikon genomics groups, for giving the opportunity to present the result. Here I leave the contacts of the company and my personal email. Thank you very much to you.
Thank you, Dr. Topazo, for sharing such a successful validation result of our NIXI technologies. And we're so pleased to learn such exciting clinical application cases in Argentina. And it has been such a great effort about delivering this product in South America for us through such a professional operation and robust result. Thank you. And we would hope to see more and more clinical and research works to supporting this advantages and benefits of our NIXI technologies elsewhere in the world. And one important thing I would like to emphasize is that the Dr. Topazo also point out several key factors and give really useful tips to our audience to pay attention to during the experiment in order to achieve success and the accuracy of the result, especially during the sampling procedures. And I hope these tips are quite valuable to other users for their validation experiments as well.
Hi. So now is our Q&A section. So if you have any questions, please feel free to ask. Thank you. And our Dr. Pai and Dr. Topazo are here. Let's welcome them.
Hello.
Yes, hi all. This is Amol from Yikon Genomics. First question is for Dr. Pai. Dr. Pai, do you recommend any specific culture media brand for NICS? May I know what kind of brand did you use for your NICS procedures?
So we are using the Quince media. As per I think Yikon had given instruction, because different media have got different compositions, and that can maybe influence the DNA amplification. So we use the Quince media, the sequential cleavage and blastocyst media. We also use it for a routine work. We don't like to use the single media. We like the sequential media. So we use the Quince as a rule.
Okay, great. Dr. Topazo, would you like to share with us which media do you recommend for NICS?
Sorry, what recommendation you say?
Which culture media?
Okay,
like
yes.
Recommend?
Well, yes, I understand. You know that we are not a company of in vitro fertilization. We basically do the second part, the genomics and the bioinformatics reporting. But in any case, the experience with most of the clinics was that most of the media works well. But here in Argentina, most of the clinic works with a global medium. Yes, this works very well. Yeah, we only have a problem with a single clinic. I don't remember exactly, but of course I will mail you privately a single media that doesn't work. But it was only a single clinics. Yes, but most of the medium works well.
Oh, okay, great. Thank you for that answer. The second question would be, is it really necessary to use laser-assisted hatching for obtaining the NICS results? And what was that in your cases?
Well, here in Argentina, we work with, as I said previously, with different companies, 14 different companies. Most of them has not laser. Then the possibility to have or to do a laser pulse is impossible. And I say that it's not necessary. We are working with clinics, only 2 clinics that has laser, and no, they don't do that. They don't do the laser pulse. But in any case, what is more important is to um, is to um, is to grow the embryos. I don't know what the Dr. Pei think about that, but to grow the embryo to day 5 to day 6, that is a well characteristic of the growing embryo. And if you do that, the laser is not important. This is the experience we have. But what is critical, what is very, very critical, is the washing day 4, because you remove most of the previous DNA that in most of the cases are DNA that are I say it's not very good. It's not a quality. Has not a good quality, and has many interference with the quality of all the tests. Then, if you use the day 4 washing of embryos and only has the day 4 for 5 or 6 growing in a new media, in a clean media, and you take this culture media, the results are very, very good, and no laser are necessary. This is the experience that we have now.
Sure, Dr. Dopazo. Dr. Pai, would you like to enlighten us with your answer?
Yeah, so I agree with Dr. Dopazo. So initially, when we started doing this procedure, we used to do the laser. Then Catherine Rakowski's paper came that day. The expanding blastocysts, day 5, day 6 is the key to the whole thing. So we started shifting, and now what we do is we culture them. Of course, this is a tedious part, because the embryologists, they wait for the expansion of the blastocysts. So we culture them, and around day 5.5, if the blastocyst does not, if we feel it's not going to expand, then we give the cut. You understand, with the laser. And so then by the time it helps to expand the blastocyst, most of the time the blastocysts are expanding, and we are also now counseling. So now what I've done is I also have an add-on counseling if there is no expansion, because if there is no expansion, the blastocyst is compact, there is a possibility that DNA may not amplify. So in those patients, we counsel them that we may do an invasive biopsy. So you have to be flexible. And one thing I would like to tell you, that because of this litigation area in Australia, we should not discard the blastocysts. It's very important not to discard any blastocyst. We have a special consent. We say that if there is an abnormal blastocyst, we will store it. If you want to discard it, we will offer them a biopsy, extra biopsy, actually invasive biopsy, to confirm the diagnosis, because there can be a potential area of litigation there. So we just hold on to the blastocyst, don't discard anything. So, and I always tell them that it's a grading thing. It's a grading, it's not diagnostic. We have the 3-4 page consent form. We tell them it's a grading technology. When you tell them that, the patient, because I'm a clinician. See, I'm a clinician come embryologist. We have done 230 embryos. We have not yet opened this technique to others, because it's a very important, sensitive technique. It can cause major. So we counsel. So my patients, I counsel them. I say, listen, it's a grading technique. It cannot be as accurate. We all know that it is better than the invasive, but the world doesn't accept it as yet. So we have to be very, very careful when we tell them that, if there is an invasion, the embryologist may not be skilled. He may damage the embryo. Then they say, no, doctor. We would like to have, we accept that it's a 80% accurate technique. I tell them that. Then they say that, yes, we accept that, and we'll use it as a grading. So I have a combination. I've got 3 artificial intelligence systems in the lab already, and which are visual. Then I've got the next gen sequencing, non-invasive system. So together, combination of it. Just today in the morning, I've done 2 transfers, single blast, and combining these various techniques, that gives a result.
Sure. Thank you, sir. So as the next question is, as you know that we tend to use mineral oil in IVF to avoid evaporation. So the question is, does that interfere in NICS results as well? And how to encounter that?
Herman,
maybe
would you
Dr.
like to?
Yeah. Dr. Dopazo, can you start, please?
Yes. Again, we as Biocodices is a company of genomics. Then most of the first parts, that is what is happening in the clinics and how the culture media is taken and how the embryos grow, etc. We basically follow the protocol of Yikon genomics. And basically, of course, it's very important not to get the oil because absolutely interferes with the amplification and the good results. But it's very easy in the clinics to remove all the oil in the sample. Yes, this is the question.
Yes, it does.
Okay.
Dr. Pai, yeah. Dr. Pai, can you
No,
please?
I think that oil, if you use any good oil which is properly washed and all, we buy oils which have got culture media in it. So they leach the contaminants into the culture media. So, these kind of oils which are there now, the newer, culture media oils are very good. The quality is very high now. I've been doing IVF for last 30 years. 1990, I started doing IVF from Melbourne. So that time I too used to make my own media. Now everything is so well done by the companies. I feel more or less, as Dr. Herman said, it's you can use any media, it doesn't matter. But I am very scared of using anything and deviating from the norm because we are in that early phase. See, it's very important that we use and handle this technique with care. We don't want bad name for the technique. It's very important. So it has to be run parallel with the standard technique. The gold standard presently is the biopsy technique. We have to nurture this technique. Slowly, we have to nurture it. We have to grow the technique. And slowly, we have to take the biopsy technique out of our armamentarium and replace it with this non-invasive technique. So it's an evolutionary process. It's not a sudden process. And that is why I would like to caution everyone. You must be very careful when you use, especially contamination. So cumulus cell dissection is very important. Maternal contamination, contamination of the embryologist, your own DNA can get contaminated. So you have to be very proper hair, covering your hair, proper wearing gloves, not having too many people in the lab, compartmentalizing the biopsy from the amplification part, from the reading part. These are all very important things because otherwise some group will say, oh, this is a bad technique. See, you have to implement it properly.
I agree with you. This we have the experience that the best results are associated with a man or a woman working alone, and all the other people assist to this in trying not to move there, and not, of course, all are protected against the accumulation of cells or wherever. This is very important that the clean media when the embryologists work doing the washes or the transfers of the embryos in different capsules, etc. This is very, very important. Yes, absolutely. Yeah.
Sure, doctors. Thank you for that. The next question is for both of you. May I know what sampling method did you employ in your IVF lab? So it was day 3, day 5, day 6, day 4, day 5, day 6, or the frozen blast? Dr. Pai, please. Yeah.
No,
yes.
Harman, we use now we don't bother. Now we are quite confident. So if they are, most of the time we'll culture the blastocysts in the fresh cycle, take them to day 5, we'll dissect them well on the day 3, put them in new. But this step of Harman, maybe I'll institute that in my lab. When I go back, I'll tell the embryologist that there's a great man, Dr. Herman, who has said that we should watch the embryos on day 4 to give better DNA. So we will try it out now. But normally we don't touch. Once we do the dissection on day 3, put them in new culture media, that's it. We'll wait till day 5, day 6. Remember, the day 5, day 6 expanded blastocysts will pick up. If you freeze the blastocysts on days 5, 6, or 7, you must transfer in a frozen embryo transfer cycle on day 5. Remember that. Or even if you freeze a blastocyst on day 6, transfer has to be on day 5 after starting the progesterone. So all the blastocysts, irrespective of the day of the freezing, because we are now waiting for the expansion. We don't intervene too much because when I was talking to one of the audiences, one of them objected. They said, "How can you say that it is non-invasive when you are using a laser?" So I said, "Oh my God, what he's saying is correct." So now I've changed my terminology also. I call it cell-free DNA analysis of spent culture media. I don't say invasive because sometimes when I do a laser cut, it may become, a purist may say that it is invasive. You use the laser. You understand? So you have to be more intelligent and minimize intervention. So now, even if we have lasers, in all our 7 IVF labs, they have lasers, but we are now using lasers lesser and lesser. We wait. Only the embryologists are a bit irritable because, they have to wait for the blastocyst to expand. Sometimes they have to wait in the evening. So that is the downside of it. But it's okay. I mean, we compensate the embryologists well for that.
Sure, Dr. Dopazu, please.
Yes, sorry, but I missed the main question.
Okay, sure. I'll just rephrase you. So may I know what kind of samples it comes to you? Like what sampling methods do they use in their own IVF labs?
No, sorry, I can't understand. What can you repeat again? Sorry, yeah.
Yeah. So sampling method means I wanted to know that there are various sampling methods. As you know, day 3 we culture till day 4, day 5. Then for the day 4, day 5, day 6, and the frozen blastocyst one. Yeah. So which generally samples comes to you for doing mixed test? Yeah.
Okay. Well, the point, our experience is that as Dr. Paz said, the best results are when after the day 4 you remove all the previous DNA and you culture the embryos in a new medium after day 4, and the blastocyst expand as far as possible. When you get this this stage, you can stop the procedure. You take the medium, and this is exactly what we receive in our lab. Okay, from different parts of the country. Okay, now another experience that we have is that sometimes these results are not good, but we have the possibility that now from these frozen embryos, yes, we can re-sampling, yes, again, to test again, yes, a result that probably has no very good, that we have some mosaics or some problems of contamination, probably, yes, that we see, and say to the clinics, please, can you regrow the embryo at least 12 hours, for example? Of course, it depends if the embryo has the possibility to grow more or not. But in all the cases, many of them, yes, the cases that we grow the embryo after frozen for 12 hours, well, the results are very, very good, very, very good. And the levels of euploidy grows in comparison with the previous one. Indeed, this is amazing, but it's very interesting because this is as the last chance for that embryo to get a result for the patient or for the clinics. Then many of this kind of resampling or from frozen embryos to obtain the DNA from the culture media, as I said, no more than 12 hours, has a very, very good results. Yes. Of course, if the biological lab works or very bad, or the contamination persists, you don't have a good result, of course. Yes. But we are improving with all the clinics in the country, and results are getting better and better. Yes. It's clear what I said?
Yes,
I would like to
sure.
add to Anmol. I would like to add that there in the papers which were there in one of the papers, what they found is the where the when they were studying the concordance, where the biopsy and the spent culture media both
Yes.
showed euploidy. That was the best result. So in fact, I am actually thinking of offering a double to the patient. Both the spent culture media as well as the
Yeah.
biopsy, both are concordant. We can say
Yeah.
we'll give you a better result. So, because that will be
Well,
better for the mosaicism.
yes, what's happening here in Argentina, many countries of the Latin Americans, that the clinic has not laser, and it's impossible to us to get the embryos, and of course the clinics are not so confident to move embryos for one lab to another or different kilometers or whatever. Then it's impossible for us to make this kind of both analysis here from the kind of clinics that we work. But of course, another very important point is that I say that the Yikon Genomics products for using invasive are very good too. Our results are better than what we previously obtained using very sick or Illumina products. Yes, it's absolutely true. Yes, there are very good results. And many of the clinics working with us with trophectoderm biopsies have they improve, yes, the transference ratios in the patients, yes, for that clinics using Yikon Genomics. That this is important to say that. Yeah. Well, thank you, Yikon, for that.
Sure. Thank you, doctor. Next question is for Dr. Yanjo. How Icon's NICs generally improves the accuracy of whole genome amplification? Dr. Yanjo, please.
Dr. Harman, how many embryos have you done till date now? How many births?
I think there might be some little bit internet problem probably. So Dr. Pai, can you let us know, like, do you find the Malbec technology better than the other techniques that people use?
I don't know about that's the molecular biology, but he keeps on saying that it is very good technique. And, I also agree with Dr. Hanman that Yikon genetic products are very good. And, we are integrating. So now, initially we were doing the biopsy from one other company, we have got the Ian Torrent platform. So we were using the library part from Ian Torrent, and, it was creating confusion. Now we are integrating everything. So all the platforms are, or the biopsy as well as the non-invasive technique is from Yikon. So, all these integration will be better. So once we have an integrated platform where we can integrate biopsy, the non-invasive spent culture media, then the products of concept test, and also the ERA test or whatever. I mean, if we can integrate all these things, expanded carrier screening, all these things together, and PGTM also, all these things, once we can integrate, see, the whole genetics has to be, we are in a barrier situation where genetics is not accessible to the whole world. We have to, and I'm sure Yikon will make it possible. China has always been at the forefront of that, and we have to see that more and more tests are done. And, so as the barrier between genetics and the general population, we have to break that barrier down. Everyone should be able to access genetics, precision genetics, when they are taking. So I foresee that 20 years from now, it will be very easy. So let us hope that we are a part and parcel and contribute to this progress.
Sure. Thank you, Dr. Pai. Now it's over to Wei Zhao. Wei Zhao, please carry on.
Hello, Dr. Pai and Dr. Dopazzo. I have also several questions I want to ask. The first is, how many media changes are recommended until the collection of the spent medium for NICS?
You say the number of media changes?
Yes, the times
okay,
of
well,
media changes.
Well, we have different experience here in the with the clinics we work. For example, of course, you know that as if you use many changes, this is well, it's costly for most of the clinics to do many changes of washes, but they are trying to minimize the number of changes. What we recommend is that they work as the protocol of Yikon Genomic said, that is at least do the two different changes before and after the day 4. Yes, the day 4 is the last change. This is the last change of washing, and previously you do at least one change of the embryos with washing. Basically, because when you do these changes of medium, you remove the maternal cells, and this is very important. We have the experience that if you work very well removing that, probably with only one or two changes is sufficient. But as in the case you use more than that, well, we obtain more better results. It's clear the point. The point here is as many washes you do, you remove maternal cells and remove contamination. The key point is the day 4 because it's the last one. Okay. And in the case that you froze the embryo and after that you get another cultivation for 12 hours or whatever from south embryos, well, you need another cultivation of the embryos, and you remove cumulus cells again if they have, and results are as much better. Is clear the answer?
Okay, thank you very much.
Okay.
And okay. I know Dr. Pai's opinion, thoughts.
I mean, sorry, Dr. Harman has answered it very well. So it's very important to dissect the cumulus cells, and we don't normally now we don't really freeze and thaw. And of course, our process is that normally we do day 3 freezing, but now when I'm doing NICS, non-invasive chromosomal screening, I don't do day 3 freezing. I do day 5 blast. So I'll just culture them in a fresh cycle. I'll dissect them on day 3, but I may add a step after listening to Herman, maybe a additional step I will add on day 4 and dissect it better. Maybe that will improve. I don't know. I'll discuss with my embryologist. But our standard technique is dissection on day 3. Very good dissection. No cumulus should be there. Then putting them in 20 microliter droplet. The volume of the droplet I feel is also very important. Not more than 20. Otherwise, there's too much of dilution. So 20 microliter droplet. Many times we also have an embryoscope. So we use the embryoscope to eliminate. So if the embryoscope shows an abnormal fertilization, then we don't have to use that blastocyst. We eliminate that blastocyst. So we so we use the embryoscope, and the embryoscope has very small droplets. 20 microliter wells are there. So we pick it up and then we tube it and we amplify it. And then we wait normally for the blastocyst to expand and hatch. And sometimes if they don't hatch, we have decided that we'll be aggressive. So in a blastocyst that doesn't hatch, we will do a biopsy. So again, I have also now divided my protocol. So if a patient is more than 35, she's in the first or second cycle, or a patient who has got repeated IVF failures, in that patients, I offer them the non-invasive. For patients with a high order recurrent pregnancy loss, I will still offer them invasive biopsy, especially if there are miscarriages and there is a balanced translocation. I would offer them a biopsy. This is one
Thank
question
you.
actually I would like to ask you, that if there is a balanced translocation in a couple and history of recurrent miscarriages, can you use non-invasive technique? What is your opinion, Harman?
I have an opinion.
Yeah, please.
I have an opinion. Well, if we did some tests using that, because when we have the results of previous embryos with structural arrangement, and when these structural arrangement are very clear, very high, that is more than 10 megabases of the arrangement, well, you can do you need more number of reads in your run. Yes, that is you can't do the same analysis as the PGTA, non-invasive PGTA, classic non-invasive PGTA using Yikon Genomics products. You need to grow, yes, the number of reads depending on the level of the megabases you have this rearrangement. But clinically, we work with invasive, yes, now, but we have some results about that, yes.
Okay.
But you need to grow the number
because
of
I had a
reads.
patient recently, and my embryologist and my geneticist said that no, it's not better to not do it. And even we asked Yikon, they said no, you need to do a biopsy. So we did a biopsy and could get the pregnancy continuing. But like yes, if they can evolve a technique for the balanced translocation non-invasive, it will be good.
Yes, it depends of the level of resolution you need to solve, and but our experience is that using just for not for clinical cases but for research, you can do that. But when the structural arrangement are P and Q arms with higher than 10 megabases. Yes, higher than 10 megabases, and growing the number of reads. Yes, basically to 2.5 or 3 million of reads. Yes, not 1 million. Yes, for each embryos. Okay. Right. Now you need
yeah,
2 or 3 slots. Yes, in the batch of the. Yeah, cool.
Yes, that's also my next question. So 2 million reads.
So, yes, I also want to comment on this. Dr. Pai, I think that is a really good question that you asked, and lots of the people that would like to consider, say, using non-invasive testing for, say, PGD. But as Dr. Gepeso suggested, I think it is still to be safe to do research only for this non-invasive at this stage. And especially it's a case-by-case scenario as well. So for example, if the breakpoint is really small, but unless you have the breakpoint very clearly pinpointed based on the previous samples or the families, otherwise it will be really challenging or risky clinical decision to proceed with the non-invasive at this stage. But we hope with the development of the technique and the cumulative cases, and then we will have the non-invasive approach for the PGD in the near future as well.
Okay, great. We are waiting for that.
Yes, we're working on it at the moment.
Dr. Harman, how many embryos have you done now in your lab? What is your, what's your experience now on this?
You are talking to me?
Yeah,
sorry.
Yeah. I was asking.
Sorry, because because okay, sorry. Well, our experience working with non-invasive, yes, is about 500, yes,
Okay. Very good.
500 embryos, 500 embryos, yes.
Very nice. We have done
Yeah, well,
230, but I have not opened it as yet. It's only my in-house. So I was, I thought I will just fine tune it before I open it to the, I'm always worried about the contamination and all level and all that. They need to be properly trained. But I think now maybe the time has come for me to open it up.
Yeah. Yes. And well, the point is that it's a very, very useful method for the clinics here in Argentina, because it's a very, well, it's not as very big as India, but it's a very big country, and most of the clinics in different parts has not laser, and then a man or a woman with a laser must transport the laser from the center of the country to one of the extremes, and must to centralize all the cases to grow in the same cycle, in the same week, etc., and spend more than a week there, and it's very, very expensive for the clinics to maintain all the system. Then for us it's very easy, because we work with a single box with the buffer that Yikon Genomics provide to us. Then we send to the clinics at room temperature, and they come for us in minus 20 degrees. Yes, and this is very easy. It's very easy. The logistic is very easy to return the culture media. And of course, the hard work is that each of the labs in the country works well with the protocol and improve the results without contamination. But the experience is that some clinics, for example, do very well in the first run. It's not the classical result, but after 3-4 runs, yes, of experience in each of the lab, we talk with them and say, be safe of this part, how we are the risk of each of the steps in the protocol to take the medium, and labs they do well in no more than four trials. That's good. Of course, sometimes there are some fails, yes, because they are it's a classical one, but we can repeat them, yes, again, and obtain. We after frozen embryos are towed, etc. We can retain.
Right. You said that you thaw them for 12 hours and then you can collect enough DNA and
yes, and again send the samples, etc. And we do again. Yes.
Sorry for interfering. Just another question. How can we improve the false positive rate and the false negative rate, as well as the Matthews correlation coefficient for the next test in the fresh embryos? Dr. Pai, could you help?
And I would, I can't comment on the lab part so much, but maybe you can tell us more about false positive, or with Herman can, but but I think that from my clinical perspective, because I'm a clinician and embryologist, I'm not a molecular biologist, but from my clinical perspective, once the protocol is in place and once we are aware of what is what, as Dr. Herman has said, it is very simple and everyone can do it. So unlike a biopsy, which is a very complicated procedure, you may damage the embryo. There's no way you can tell in a biopsy whether you have damaged the embryo or not. It's very difficult, There's no parameter by which you can say, okay, this embryo is damaged or its implantation potential has gone down. So I always tell my patients, see, for 30 years, did we do any PGTA? No. Did the body do any PGTA? No. So this is a new technique. So why go for the extreme technique? Let's go for the middle technique, so it gives you a good amount of knowledge, and you have to be ready for accepting that it is not, though we know that it is very accurate, but when we are counseling the patient, we have to tell, okay, it's quite good. So then when you do that, they are very happy with it, because you cannot guarantee that there will be no miscarriage. And I always tell them that you have to do a chorion vellus sampling or a NIPTS, as please, non-invasive prenatal testing. Once the patients conceive with this test, we must do a NIPT, I feel. So, just to be on the safe side, or, that way.
Yes, thank you. I understand that. Thank you very much for your comments and suggestions. And I have no other questions for now. Do you have any?
I think we can maybe get all of you on the screen so we can have a photograph of everyone together, the participants.
Yeah, and thank you, Dr. Pai, and thank you, Dr. Dupozo,
Yes.
and thank you, Dr. Zhou. Thank you all.
Thank you.
Thank you
so
so
much,
much.
Dr. Pai and Dr. Chapisa, for your great talk today. We're all really excited about your latest progress, and we're hoping that together we can move this technique even further and benefit a lot more users worldwide. And also we will remember that the aim for the non-invasive testing for the PGD to get it solved for the clinical needs in the near future as well.
Thank you. Thank you very much. We had a great time, and I really, as Dr. Harman was saying, the Chinese are very good in their science, no doubt, and their manufacturing, and the products are very good.
Yes, yes, I agree.
Thank you,
Yes,
Dr.
thank you.
Thank you.
Thank you, Dr.
Thank you,
Thank you.
Dr.
Thank you.
Dupozo and Dr. Pai.
Bye. Bye. See you.
Bye.
Good night.
Appreciate
Have a good day.
Have a good day.
Bye.
Bye bye.
Bye. Hope the
Bye.
audience are enjoying this session as well, and we will see you in our next session. Goodbye now. And have a
Bye.
good day,
Bye.
everyone.
Bye.
See you next time. Yeah. Bye bye.